Hi, everyone. I'm Pangkong Fox, the Science Engagement Director for the CACNA1A Foundation, and I'm here today with Dr. Fikri Birey, who was awarded a $57,000 grant from the Million Dollar Bike Ride Program through the Orphan Disease Center and from the CACNA1A Foundation. These funds will support his work that's focused on the molecular and functional mechanisms underlying cortical activity in CACNA1A-related epilepsy. Dr. Birey received his PhD from Stony Brook University and did his postdoc at Stanford. And he is currently an assistant professor in the Department of Human Genetics at Emory University, where his lab focuses on understanding the molecular events that help to guide human brain development using organoids as a model system. So congratulations, Dr. Birey, and thank you for taking the time to talk to us today.
Thank you for having me.
Okay, so before we discuss the work that the grant is funding, could you briefly tell us how you became interested in studying CACNA1A? I know that you had worked on CACNA1C, another calcium channel prior.
Yes, I indeed worked on CACNA1C and CACNA1C related disorders, and that was during my postdoc in the laboratory of Sergio Pashka at Stanford, and that's really where my interests around calcium channelopathies, this umbrella term for calcium channel related genetic disorders, really emerged. Um, When I joined the lab, the Pasca lab, I was really interested in developing these human cellular models to really better understand these diseases that have their origins in brain development. And the first set of models that I developed, I ended up applying these models to investigate this severe neurodevelopmental disorder that the Pascal lab had been studying for over a decade at that point, called Timothy syndrome. And this is a syndrome that is caused by mutations in the calcium channel gene CACNA1C, which is a similar gene to CACNA1A. And similar to mutations in CACNA1A, the mutations in the CACNA1C can really affect brain function and leads to autism, epilepsy, among other syndromes. And through that work, applying our new models to understanding Timothy syndrome, I really became fascinated by calcium channels in general and how genetic perturbations in these genes that encode for these channels can really affect neuronal function at such a fundamental level, and how even small disruptions in their function can have really widespread effects. And when I was looking to start my lab here in the Department of Human Genetics at Emory, I was having an on-site visit about like late 2021, where I was meeting with a number of faculty and clinicians here, and I was giving talks about my Timothy syndrome work. And I was having dinner one evening with a group of clinicians and faculty, and I got to talk to a clinician and now a colleague more in depth about our work. And what she ended up telling me was that there was this cohort of patients that were coming through the Emory system, patients with CACNA1A mutations. And I already knew when I started my lab, I wanted to continue working on calcium channelopathies. And the fact that these patients were being seen right here at Emory also made the work feel somewhat more urgent and kind of relevant to the community I was looking to join. But I gotta say, what really solidified our long-term interest in CACNA1A was our interactions with the foundation, with the CACNA1A Foundation, where we really got embedded in this network of patients and patient families and like-minded researchers that are all very extremely driven and passionate about really understanding and hopefully one day eradicating this disease. And that really to this day really drives a lot of the motivation on my end and my team's motivation. And we're really, really excited to be a part of this dedicated effort for years to come.
That's a great story. And, we're excited that you're part of our research network as well. We're certainly happy that you decided to have dinner with that clinician and hear a little bit more about the CACNA1A community. So yeah, that's wonderful. So before we get into some of your research, can you help define what an organoid is for our families and how it can be used to study what's going on inside the brain?
Absolutely, yes. So brain organoids are fundamentally these three-dimensional models of the developing human brain that we grow in our labs in a dish to study the processes that are critical for brain development and how these processes unfold in health or go awry in disease. This brain organoids, we generate them from what's called, it's a bit of a mouthful, induced pluripotent stem cells. And these stem cells are generated through a process called reprogramming, where we basically take a small sample of a person's skin or blood and really turn them back into stem cells. And what's so special about the stem cells is that they have the potential to turn into almost any type of cell in the body, and that includes brain cells. So once we have the stem cells, once we have the skin cells or the blood cells, then we program and then we have the stem cells. What we can do is we can really guide them towards forming these three-dimensional structures that end up resembling very specific parts of the developing brain. And we have done now years of work describing different protocols where we can generate organoids resembling the cerebral cortex, the thalamus, the midbrain, the brainstem. Some of these regions you might have heard related to CACNA1A related disorders as well. And not only we can make these organoids separately, and study the regions in isolation, but we can also assemble them, put them together. And what this enables us to do is to really study the interactions between these regions that arise during early brain development. And these models we have come to call assembloids. So in our arsenal, we have brain organoids, brain assembloids. And what makes these organoids and assembloids really, really powerful tools is that they carry the genetic makeup of the person that they come from. And what that means is that if a patient has a mutation in CACNA1A, for example, we can create a model of their own brain development in a dish and study how this mutation can affect their neurons. And a nuance there that is also very important, how this mutation affects their neurons with all of the genetic landscape, all of the genetic uniqueness of an individual that might also contribute to the nuances of how a certain disease mutation might manifest as well. Yeah, so we are overall very excited about the potential of these models, and we are excited about them not because they're we've come to appreciate them as novel and effective tools to understand new disease biology, but also this newly emerging platform to test new genetic therapeutics. Last year, we reported, for example, a new brain organoid-based antisense oligonucleotide screening platform, where we were able to identify about up to 4 different ASOs, this antisense oligonucleotide small molecules that can tweak gene activity. And we were able to, with this shortlisted 4 ASOs, we were able to genetically reverse many of the disease phenotypes that we had described in Timothy syndrome. So we are very excited in general, as I said, about the potential of this model. And what we're really hoping to do is take what we learned in Timothy syndrome and bring all of that knowledge and know-how to really understand CACNA1A related disorders better and potentially develop novel therapeutic approaches as well.
Mhm. Yeah, you bring up a good point there in terms of the fact that there's a lot of really great work and experiments that could be done in these 2D model systems, such as these mammalian cells or even iPSC neurons themselves. Those are really flat in grown on a plate, and you can do great work. But we have to remember that our brains aren't flat cells, right? Our brains exist in this really complicated network, and there are, can I say billions of neurons in our brain, right? And trying to understand, and the point isn't to go and understand what each one of them is doing, but which ones are really the underlying cause or the origin of the disease that we're studying. And so having these tools at hand, having these organoids and then assembloids to study this it really is groundbreaking.
Absolutely.
Okay, so let's get to the fun part. Could you please summarize the research project that the CACNA1A Foundation is helping to fund, and explain that to our community, and explain how you envision this work moving our community towards specific treatments?
Sure. So the work that is now being supported very generously by the foundation is really focused on understanding how different kinds of CACNA1A mutations can lead to certain common outcomes, such as epilepsy or seizures. As we all know, CACNA1A mutations are quite varied in how they affect the function of the channel. Some of them make the channel overactive, what we come to call these gain of function mutations. Others make it underactive, also function mutations. We sometimes refer to these as what, and with some others that are a lot more sophisticated and more nuanced changes. Yet, despite these opposing or divergent effects, there are many patients that experience this common syndromes like seizures or epilepsies, which can be quite debilitating, independent of what kind of mutation. So with this study, what we're trying to understand is this question of how this very different disruptions in calcium channel function due to these different mutations might lead to seizure-like states, these hyperactive states in our organoid and assembloid models. And really the core motivation that drives this question and our kind of drive to try to answer it is based on our belief that by uncovering this different mutation-specific pathways that might uniquely contribute to an epilepsy-like state, we can really inform more targeted and maybe scientifically guided approaches to a new family of therapeutics where a cleaner, maybe more specific, and then maybe more effective and maybe specifically tailored towards CACNA1A epilepsy. So to address this question, basically we are using, as I said, our organoid models and assembloid models, and we're combining it with several state-of-the-art techniques. And the assembloids and the organoids that we are driving, we are generating, are basically derived from the stem cells that have either the gain of function mutations or loss of function mutations. So once we generate these organoids from the stem cells, then we end up generating these models, assembloid models that have these specific mutations, and develop these neuronal networks in a dish, with this genetic background. So using these models, we're basically, as I said, to kind of tackle this a bigger question of mutational heterogeneity and shared outcomes, we're kind of breaking this down into 3 sub-questions, let's say. The first question we're asking is, how are these mutations really changing the way neurons communicate in assembloids? And we're basically using advanced microscopy and certain electrical recording techniques to see how neurons are firing and how neurons in networks also are firing. We're interested in how individual neurons behave, but how the network itself, where the networks of connected neurons are also behaving. And we think just by following the emergence of these networks in gain of function and loss of function assembloids, we're going to be able to pinpoint how these mutations are shifting the activity of very specific cell types in neuronal networks, and how the disturbances in specific cell types might be contributing to differences in how these networks are engaged. So we're hoping to see that the ultimate end route to the status of the networks will be hyperactivity, but that might be a consequence of too much activity in one cell type and too little activity on the other, or just uncoordinated connection and connectivity or communication between different cell types that make up this network. So we are hoping to gain that level of granularity or resolution into not just saying there's more or less activity, but really figuring out the players here. So that's our first goal. The second goal is to try to understand the genes that might be driving these alterations in the network activity. And for this, we're using a technique called single-cell RNA sequencing to see which genes might be turning on and or off in response to these different mutations, and how this changes in gene activity might tell us something about the assembly network activity. And there we're hoping to connect that molecular view of things with the neuronal functional view of things. And really our third aim is more of a translational aim, where kind of inspired by the work that we've done in Timothy syndrome, we're asking specifically for a very severe gain of function mutations, A713T, that lead to early onset, very severe forms of CACNA1A epilepsy. We are asking whether we can develop antisense oligonucleotides that could fine-tune the activity of CACNA1A, and whether that would serve as a genetic rescue of some of the phenotypes that we're hoping to uncover in the first 2 experiments. So this is the kind of the outline of the work, but I really want to finish answering the question with another question of why does this all matter, right? So this is what you were getting at also. And right now, as we all know, there's no targeted therapies or treatments for CACNA1A disorders. We are borrowing from therapies from other conditions and then hoping they help. They help sometimes, but they don't other times. Here, by kind of getting a clear picture of what's really going on, both at the cellular level, the network level, the molecular level, really dissecting things down and connecting them to individual mutations. What we're really hoping to do is that we can start developing approaches that are tailored to CACNA1A related disorder itself. And this could be ASOs, but this could be small molecules or other future interventions with ASOs, but it could be a number of different things. But we think really understanding the underlying biology of the disease could really lay the foundation for therapies, future therapies could really address the root cause instead of treating the symptoms in future.
Yeah, thank you. I mean, this is a really comprehensive project too, but sort of in a nutshell, you're really trying to get into the nitty gritty of how each of these mutations not just impact the calcium channel itself, but the activity and then how that activity impacts the greater neuronal network. And then understanding what other players also, factor into that, because we sometimes forget that, again, these genes don't exist in a vacuum. And so in any sort of disease, pathology, there's not just the targeted gene itself, but there are all these different molecules that play a role in that. And by understanding what some of those other molecules are, in some ways you can find new targets that would still directly impact the pathway that you're looking for.
Yes.
So yeah, this is really exciting that you've got everything sort of laid out, and fingers crossed that it goes,
Yeah.
goes as planned, because we know
Yes.
science can sometimes
I
make you pivot a little bit, but
Exactly. The kind of science that we do in the lab, we sometimes think of as them as dominoes. We like, or like the dots that we like to link. That's why a lot of what we do is trying to connect the molecular to cellular, cellular to network dots that we and then here also we think there are different lines, different routes, right? So different dominoes. So you can line the dominoes up in 3 different routes, and then they might start out different, but we think that there's a point where they might be converging, right? There's gain of function line of dominoes, loss of function line of dominoes, and then by being this model, what it does is really helps us kind of be able to track all of these levels of regulation, so that we can track the dominoes falling and really connect those dots, and then we can really see how something that might be related to patients like hyperactive networks might have their root causes where they might be starting from, and how this different mutations might be converging on later on. Yeah.
great. I know that you kind of touched on this before, but what excites you the most about working with the foundation and the CACNA1A community? And how has meeting families really impacted your work?
Yeah. This has been a very transformational part of the science that we do. Just meeting either through the meetings, or one-on-one emails, or our other interactions that we have with the patients through the foundation, meeting them, hearing about their stories, really seeing what these conditions look like beyond the lab, and all the daily challenges, the uncertainties. It really changes the perspective, and and it has definitely changed mine. And there's a sense of urgency with this kind of work, especially in all of our disease modeling work, but just because our kind of a connection to the foundation, it really has colored our science with a unique sense of urgency, I would say. And the whole community is incredibly engaged and knowledgeable and passionate. Every time we meet, I learn more, and I just feel extremely lucky and inspired to be a part of it. And I feel like this connection to the families and the community as a whole has made an impact, not just on me, but on my team. So just seeing it through also eyes of my trainees that also participate and find new meaning in their work through the eyes of the patients, and then the other advocates in the foundation. So yeah, I mean, we are scientists. We're interested in the biology. Obviously, we get just calcium channels in general is interesting to me from a biological perspective. But I feel like what really drives the work day to day is our kind of motivation that is empowered by all of you to really find real world solutions. So to you know to the things like CACNA1A epilepsy and really try to do this so that we can have impact in a as soon as we can. Yeah.
Yeah, no, that's great to hear. And I hope that you understand how much our families, especially myself too, being a CACNA1A parent, how much we appreciate your dedication. And I'm glad to hear you echo, we've had other basic researchers say the same thing, that meeting a family for the first time, seeing how this disease is out of the lab is it really opens up your eyes, but it also opens up the eyes of the families when they see researchers who are willing to take that chance and study a disease and meet the families and get inspired that way too. So, well, congratulations again, Dr. Birey, on receiving this grant. Thank you so much for your focus on and your dedication to understanding, again, all these molecular and network reasons behind CACNA1A related disorders. We're excited to see the data. I'm excited to see how the dominoes fall and how they might converge on something and how it can lead to new treatments for, down the road for the CACNA1A community.
Likewise. Thank you so much.